All right, we're going to get started. A tour of the cell. And that's a really, really close up with a really specialized microscope of a cell. So the microscopic world of cells. Organisms can either be single-celled, such as most prokaryotes and protists, which are terms we'll go over in a little bit, or multicellular, like plants, animals, and most fungus. So there are some living things that are only made of one cell. They're called unicellular organisms. And prokaryotes and protists are examples of those unicellular organisms. Everything else is multicellular, just means made of many cells. Cells were first described in 1665 by Robert Hooke. By the mid-1800s, the accumulation of scientific evidence led to the cell theory, which states that all living things are composed of cells and all cells come from other cells. Light microscopes can be used to explore the structures and functions of cells. When a scientist examines a specimen on a microscope slide, light passes through the specimen and lenses magnify the image. These are the kind of microscopes that we would be using if we were in class. As we are not, your lab this week is virtual microscopes. Figuring out what a microscope looks like, what the parts are called, what you can see when you look through it. You guys are doing that all virtually. And that is a light micrograph or microscope of a protist, a paramecium. It's one of our single-celled organisms that exist on this planet. Electron microscopes are another tool to look at very, very small things. They're very expensive and most high schools don't have them. We just have the light microscopes. But it uses a beam of electrons, which results in 100-fold better resolution than a light microscope. There's two kinds of electron microscopes, and they reveal different parts of the cells. There's a scanning electron microscope for viewing surface features. On the left, that's still a paramecium, but it's the outside of it. You can see all the little hairs on it. They're called cilia. Helps it move from place to place. And there's a transmission electron microscope for viewing internal structures. And you can tell that picture is a lot more detailed than the picture on the previous slide. The electron microscopes are usually used at research facilities. The most powerful electron microscopes can magnify up to 100,000 times and distinguish between objects that are 0.2 nanometers apart, which is very, very, very small. On the left, you can see the eye of an insect in great detail. And in the middle, I believe those are salt crystals. That's what salt looks like, very, very highly magnified. Two major categories of cells: prokaryotic cells. Like we said, these are the unicellular organisms like bacteria and archaea. Everybody's heard of bacteria. Archaea is a little more obscure. And then there are eukaryotic cells. This is what multicellular organisms like us are made of. Protists, plants, fungi, and animals. We will have chapters dedicated to all of those things, or at least sections of chapters. There's a whole chapter about protists. There's a whole chapter about plants. And then fungi and animals kind of get a little grouped together because they're eukaryotic cells that are Multicellular. All cells have several basic features. They are all bound by a plasma membrane, a thin layer that just holds everything inside. Inside all cells is a thick jelly-like fluid called cytoplasm, where the cellular components are suspended. If you think of a Jell-O salad, the Jell-O it's hard, but it's not too hard. It's kind of wiggly, but it holds like all the fruit in place. That's what the cytoplasm is like. All cells have one or more chromosomes carrying genes made of DNA. And all cells have ribosomes, tiny structures that build proteins according to the instructions from the DNA, or even the RNA, because some organisms are made of RNA. Prokaryotic cells are older than eukaryotic cells. Single cells evolved first and then kind of bunched up and grouped up together to become eukaryotic organisms. Prokaryotes appeared approximately 3.5 billion years ago, when they evolved to perform the process of photosynthesis about 3 billion years ago. We'll talk more extensively about that when we get to plants. Once there was enough oxygen in the atmosphere, we call it the Great Oxidation Event, eukaryotes were finally able to evolve. Eukaryotes need oxygen to live. And this happened about 2.1 billion years ago. So when we talk about things evolving, we talk backwards. How many million years ago something happened or something evolved? That's kind of a timeline in the picture. And the oldest stuff here is on the top, 4.6 billion years ago. As it goes down, things get younger. Prokaryotic cells are usually smaller and simpler in structure than eukaryotic cells. A prokaryotic cell lacks a complete nucleus. Its DNA is coiled into the nucleoid region, which just kind of floats in the center. You can see at the bottom picture. There's the nucleoid region, the red squiggly in the middle. But it's not separated from the rest of the cell by its own membrane. These cells also have cell walls outside of their cell membrane, just like plant cells. So plant cells and prokaryotic cells have cell walls. Eukaryotic cells are all fundamentally similar, which means a cell in a human has the same features as a cell in a cat. We have a lot of the same features as a cell in a fungi, a mushroom. The entire region between the nucleus and plasma membrane is the cytoplasm. And the cytoplasm consists of various organelles suspended in liquid cytosol. So the cell on the bottom is a very, very simplified cell, just to show you. Cell membranes on the outside, the nucleus is in the middle, the dark blue, and everything else, the light blue, is going to be the cytoplasm. And all the organelles we're going to talk about are suspended and stuck in that cytoplasm. Eukaryotic cells have organelles. They're like mini organs, membrane enclosed structures that perform specific functions. They're embedded into the cytoplasm and has the consistency of jello, like I said. So this is a more realistic picture of a cell. That dark spot in the middle will be the nucleus. All the other little structures are the organelles. And all the gray in between, that's the cytoplasm that everything's stuck in. The plasma membrane, it can also be called the cell membrane. You'll see it both ways. It separates the living cell from its non-living surroundings. So if you think about what do we have on us that separates our insides from our outsides? Anybody think of anything? No? All right. Skin, good, Christian, yep. A cell membrane is the skin of the cell. For us, it keeps all of our muscles and organs and stuff in, that keeps bad stuff out, or it's supposed to, which it does most of the time. For the cell, it's the cell membrane. All right, all cells have cell membranes. Prokaryotic, eukaryotic, plant cells, animal cells, all cells. Some cells have that extra layer of cell wall, but all cells at least have a cell membrane. The function, like we said, it's a barrier that separates the inside from the outside, keeps organelles in, keeps other stuff out. And up close, this is what it looks like. It's not just a solid wall. It has to let some stuff in and out. Our cells need oxygen and water, nutrients to survive, so it needs to let some stuff in and out. But just some stuff, not everything. Its structure is called a phospholipid bilayer, which is a mouthful. But you just break it down. Phospho means part of it is made of phosphate. Lipid means the other part is made of lipids. Lipids is one of our 4 main macromolecules that we talked about last week. Bi, like a bicycle has 2 wheels, bi means 2, and then layers like a cake. So a phospholipid bilayer is the structure of the cell membrane. So make sure you draw and label this. You will have questions on it on your practice quiz and on your exam this week. But one phospholipid, this is what it looks like. You have heads and tails. The head is the part that's made of phosphate, and it is hydrophilic, meaning it likes water. Another word we use for hydrophilic is polar. Water is a polar molecule. It has certain charges. It has one side positive and one side negative. It has 2 poles. And the tails are the part that's made of lipids. If you remember, lipids are fats, and they do not like water. You mix oil and water, oil is a lipid. It does not mix. So it's called hydrophobic. And the opposite of polar is nonpolar. So a few different words we use to describe the cell membrane and its phospholipids. The fluid mosaic model. Scientists use this model to describe the membrane. Fluid means it's flexible. The two layers kind of move against each other. And then mosaic, somebody looked at it and thought it looked like a work of art with all the different molecules making a pattern in it. So they named it the fluid mosaic model. In close-up structure, there's other molecules, as you can see. There's proteins to move materials in and out. We'll talk about transport in and out of the cell, and we'll get more in detail about proteins, I think, next chapter. There's carbohydrates. They're ID tags to tell the different cells apart. So your eye cells have different ID tags than your brain cells, than your stomach cells. Your body knows where these cells are supposed to be, and if they migrate or wander to a different place, then your body will send a signal that something's wrong. And then cholesterol weaved in and out, kind of like knitting to make it stronger and make it stick together a little better. Selective permeability is another characteristic of the cell membrane. It's also called semi-permeable, so those two terms mean the same thing. You'll see it worded both ways. Semi means part or half. Permeable refers to what can or cannot pass through the membrane. The cell membrane is said to be semi-permeable because it only lets certain molecules in or out of the cell. You can see in the picture, you have the cell membrane kind of sideways. The outside, you got blue bubbles and you got orange ovals, but only the blue bubbles can pass through. And it normally has to do with size. The blue bubbles are probably water, and our cells need water, so they let water molecules pass through freely and get inside. But the orange things, whatever they are, they're too big. Cell surfaces. Plant and fungal cells have rigid cell walls surrounding their cell membranes. Their cell walls are made of cellulose. Remember, anything ending in OSE is a sugar. It protects the cell, it maintains the cell's shape, and it maintains water balance. Plant cells have to retain a lot more water than our cells do. So that cell wall helps the water stay inside the cell and not seep out. Fungal cell walls are made of chitin, but they perform the same function. However, they are all eukaryotic cells, animal, plant, fungus. They're all eukaryotic cells that each just have a few different features. Communication. Plant cells are connected by channels that pass through the cell walls into the cytoplasm of neighboring cells. Those channels are called plasmodesmata. This allows water and other small molecules to move between cells, coordinating the activities of the whole organism. Animal cells secrete a sticky glue-like substance called the extracellular matrix that holds our tissues together. And the surface of most animal cells contain cell junctions, which are structures that connect cells together into tissues, allowing them to function in a coordinated way. The This is especially important in organs like the heart. Your heart cells have 1,000,000 different cells, but they all have to beat as one. They all have to perform the same function. If any of your cells are off and they don't beat at that same time, then bad stuff happens to the heart. All right, we're going to go through all the organelles. The nucleus, perhaps one of the most important, is the director or captain of the cell. Genes in the nucleus store information necessary to produce proteins, and proteins do the actual work of cells. Our bodies would not function without proteins. But our nucleus tells the cell how to make those proteins. The nucleus is separated from the cytoplasm by a double membrane called a nuclear envelope. But it is not solid, just like our regular cell membrane. There's pores in the envelope that allow materials to move between the nucleus and the cytoplasm. And the nucleus contains a nucleolus, where ribosomes are made. So the whole thing is the nucleus. You can see there's little holes throughout its membrane, and then in the middle, that dark spot is the nucleolus. A lot of people on quizzes and tests get the nucleus and the nucleolus mixed up. Make sure you know the difference between them. Function. Stored in the nucleus are long DNA molecules and associated proteins that form fibers called chromatin. Each long chromatin fiber constitutes one chromosome. And the number of chromosomes in a cell depends on the species. We have 46 chromosomes in most of our cells. Except for our gametes, our sex cells, then we have 23. Our body cuts them in half to get passed on to the next generation. We will go over that in more detail in the next chapter, or the next few chapters. Ribosomes are responsible for protein synthesis. Ribosome components are made in the nucleolus, like we said, but they are assembled in the cytoplasm. So they're made in the nucleolus. They have to get shipped out through those pores to be able to become whole and perform their function. Ribosomes assemble proteins while in different places in the cell. Sometimes they're suspended in the fluid of the cytoplasm. Excuse me. Sometimes they're attached to the outside of the nucleus or an organelle called the endoplasmic reticulum. So ribosomes function in a lot of different places in the cell. They don't just stay in one place like the nucleus. The endoplasmic reticulum, which is a mouthful, so we abbreviate it to ER, is one of the main manufacturing facilities in a cell. It produces an enormous variety of molecules. It's connected to the nuclear envelope, and it's composed of smooth and rough ER. You can see in the picture, that big space in the middle is where the nucleus would be. And the endoplasmic reticulum comes off the nucleus, makes kind of a series of channels. and has a bunch of little dots all over it. The rough ER refers to the ribosomes that stud the outside of the portion of the ER membrane. These ribosomes produce membrane proteins and secretory proteins. Secretory means they secrete something. Some products manufactured by the rough ER are dispatched to other locations by transport vesicles, which are sacs made of the membrane that just bud off from the ER. And you can see in the GIF the little circles in between that are traveling from place to place. Those are the vesicles. Smooth ER lacks ribosomes, so it looks smooth. It produces lipids, including steroids, and helps your liver detoxify circulating drugs. So all of our cells have smooth ER, but our liver cells, where we detoxify things in our bodies, have a little bit more smooth ER, a little bit more active smooth ER. Just like our heart cells have a lot of mitochondria, which we'll get to because they need a lot of energy because they constantly beat all the time or we die. The Golgi apparatus receives proteins from the ER to repackage, store, and ship out to the rest of the cell. So they call it the post office or the UPS or the FedEx of the cell. And you can see in the little cartoon, proteins come in, they get packaged up, and then they get shipped out. Lysosomes. Only animal cells have lysosomes, which are bubbles of digestive enzymes surrounded by membranes. One of their functions is to destroy harmful bacteria. They also break down damaged organelles, and they sculpt tissues during embryonic development, helping to form structures such as fingers. So sometimes our cells or their organelles get sick and tired and worn out. Our body needs to destroy them before they make us sick. This is the job of lysosomes. And it means it sculpt tissues when we're in the embryo. Babies are in an embryonic stage. Their fingers and their toes are webbed. Our bodies get rid of that web by lysosomes. Lysosomes come here and destroy all the webbing because we don't actually need it. Vacuoles are large sacs of membranes that bud from the ER, Golgi apparatus, or plasma membrane. And its main function is storage. It's like a garage. Plant cells have a large one called the Central Vacuole because they have need of a lot more storage than us. And then animal cells have several smaller ones. The central vacuole stores organic nutrients, absorbs water, and may contain pigments or poisons. A lot of plants are poisonous. And that is where the poison would get stored. Energy conversion. Cells require continuous energy supply to perform the work of life. Two organelles act as cellular power stations, the chloroplasts and the mitochondria. Chloroplasts are unique features of plants and algae. They're the place where photosynthesis happens, where light energy is converted into chemical energy. And we do not have these. Our cells do not perform photosynthesis. So these are only found in plant cells and algae. Algae are like the plants of the ocean, of the sea. However, they're not technically plants. They are protists, which we'll have a whole chapter about. Mitochondria and chloroplasts contain their own DNA, which encodes some of their proteins. This DNA is evidence that mitochondria and chloroplasts evolved from free-living prokaryotes in the distant past. And there's a lot of different links there with a lot of different videos going into more detail about this. And we'll talk more about it when we get to evolution and plants. Cytoskeleton is a network of fibers extending throughout the cytoplasm. It provides mechanical support to the cell and helps the cell maintain its shape, just like what our skeleton does for us. Cytoskeleton provides anchorage and reinforcement for many organelles. Our skeleton anchors our muscles. So you think cytoskeleton, it does the exact same thing as our human skeleton. The cytoskeleton contains several types of fibers made from different proteins. Microtubules are straight and hollow tubes that guide the movement of organelles and chromosomes. Intermediate filaments and microfilaments are thinner and solid. Our muscle cells are made of a lot of microfilaments. Cilia and flagella are structures that aid in movement. That's what we saw, the paramecium, like on the bottom with all little hairs all over it. Those are cilia. Flagella propel the cell through their undulating whip-like motion. And then cilia move in a coordinated back and forth motion, like they're swimming. There's a lot of them, so they have to coordinate. And they have the same basic structure, but cilia are generally shorter and more numerous than flagella. The typical structure that has a flagella is like a sperm cell. The little whip-like tail is a flagella.